Dear student, today we will discuss diagnostic PCR and how it is different from the normal PCR and what is the difference between PCR and replication. So first I'll be discussing the diagnostic PCR, which is known as real-time PCR. PCR stands for polymerase chain reaction, which is also known as RT-PCR. This is also known as PCR quantitative assay or quantitative PCR. Now, this type of PCR is used as a diagnostic test, and it is widely used in case of COVID diagnosis. This is also known as quantitative PCR or qPCR, as the technique is used to quantify nucleic acid, either DNA or RNA present in the sample. Now, what is the basic principle? RT-PCR monitors the amplification of target DNA or RNA segment during PCR reaction. The monitoring relies on the use of fluorescent dye. So while the PCR is going on side by side, the monitoring is also going on, and this is done only by use of fluorescent dye such as cyber green SYBR. The amount of fluorescence is directly proportional to the amount of replicated target DNA. So when the PCR is done, the DNA is replicating, okay, and at the same time the amount of fluorescent dye also increases, so fluorescence ultimately increases. The method is rapid, quick, reliable, and perform easily with less chances of error. Now, there are certain things which you must know. PCR, the normal PCR, is the conventional technique to amplify the DNA for many purposes. Like we use it for the forensic purpose, we use it for the rDNA technology, we use it for the research, and so on. Real-time PCR, RT-PCR, or qPCR is the diagnostic technique and detect the specific target DNA amplification and quantify it. One can also tell how much target DNA is present depending on how much fluorescence is there. There is RT-PCR, reverse transcriptase PCR, performed when the genetic material is RNA. Since the RNA hold poor stability, so it has to be converted into cDNA in the PCR, and this is known as reverse transcriptase PCR. So if you see, if I talk about in case of COVID, we use both RT-PCR. I'll just discuss the methodology so that you'll come to know the different terms that we have used. Now the sample is collected from the patient, and the sample consists of course, if we talk about the virus, it consists of virus cells, not cells, but it consists of virus particles, and these virus particles are lysed, and from them whatever RNA is the genetic material, so that is released. So many chemicals are added to do this. Then out of those things which are released, it may contain protein and other things. So the RNA is purified by a specific technique, and then whatever RNA we have, we perform RT-PCR with that RNA. So the RNA which is extracted is now converted into cDNA, that is complementary DNA. So again, we do this in the PCR machine. The reverse transcriptase is added to the RNA. You add the reverse transcriptase enzyme, you add all the nucleotides, we add the polymerase, and we add the buffer, and then what will happen? The reverse transcriptase will synthesize and the DNA on RNA, and then DNA RNA hybrid is formed. Then we carry out the denaturation simply by changing the temperature, and the two strand got separated. And after you change the temperature, so RNA is degraded, and even the reverse transcriptase is no more functional. And then whatever DNA strand is there on that, another DNA is synthesized by using the primer and polymerase enzyme. So this is how you obtain the cDNA. So if you see the reaction mixture, what all things we add to the RNA in the test tube. We add buffer, add nucleotide, we add reverse transcriptase, we add primers, both backward and forward. Probe fluorescent level with cyber green. So probe is added, and tag polymerase. So probe is the actually sequence of DNA which is complementary to the specific target DNA, and it only recognizes the target DNA. Okay, and this probe is artificially synthesized, and to this we tag the fluorescent dyes or the molecules. Okay, now, once the double-stranded DNA is obtained, we will continue with the PCR cycles. So, thermocycles are there. 95 degree for denaturation, 58 degree for annealing of forward primer. Okay, and then we have for the extension temperature depend on DNA polymerase. Which DNA polymerase are we using? Okay, so each cycle consists of denaturation, annealing, and extension. Okay, and you can look at here. So this double stranded DNA, so once again we perform the denaturation, two strand gets separated and they are linked with the primers, and the probe is added which will link to the target DNA. So the marked area is the target DNA, and it will only link to the target DNA as probe is attached with the fluorescent dye. So at 3 dash and 5 dash end, there are reporter molecule which are the molecule of dye, and quencher which absorb the fluorescence. Okay, so both help in generating the fluorescence. So now when the DNA polymerase reaches probe, see, it cleaves the probe and separate the dye. So what DNA polymerase is doing? DNA polymerase is moving from here to here, and then it recognizes the probe. So whenever it comes to the probe, what it is going to do, it is going to release this molecule. Okay, these molecule, and once this is released, it produces the fluorescence. So when the DNA polymerase reaches the probe, it cleaves the probe and separate the dye. With each PCR, more dye molecules are released, and this increases the fluorescent intensity. Now, from the small amount of sample, large amount of fluorescence is emitted. So by quantitating the amount of fluorescence and plotting it against the PCR cycle, the diagnosis of a person is done. If the fluorescence does not change with cycle per time, the person is negative. The fluorescence increases with cycle per time, the person is said to be positive for a particular test. Okay, now we will see the polymerase chain reaction, the simple, or you can say the normal polymerase chain reaction. This is also known as molecular photocopying. So we make many copies of DNA in this. Polymerase chain reaction is actually in vitro replication. By this method, several copies of desired DNA is obtained, and this is developed by Carey Mullis in 1980, and it is fast and inexpensive technique to do the DNA replication. So the requirement is tech polymerase, primers, nucleotide, and buffer. So buffer provide the stability. We have different nucleotide which are added as a building block. Primers are added forward and backward, and tech polymerase is isolated for thermus aquaticus, which can tolerate or withstand 90 degrees Celsius temperature. At this temperature also, this is not degenerated, or you can say denatured. Okay, now what is the principle? PCR is a technique in which the exponential amplification of fragment of DNA is obtained. It is performed in PCR machine which is known as automated cycler, in which each thermal cycle amplifies the DNA to double. So what are the steps? Let's see. We have target DNA. So this is a specific tube that we use for the PCR machine, and this is added with all the thing. We add buffer, we add nucleotide, tech polymerase, primers, everything. So this has a double stranded DNA, then it undergoes denaturation at 94. The two strands are separated at 54. Now PCR cycle is there, so automatically these temperature are adjusted. After some time, 54 degrees Celsius annealing will occur, annealing of primer, and then at 72-degree extension, that is the polymerization occur by the tagged polymerase. And this is how from two different strands, four different strands are synthesized. Okay, and then if you repeat the cycle again, then many copies of DNA are generated. So it depends how many copies we want. We will continue the PCR to that much time. Okay, now how does PCR differs from replication? If you look at PCR, which is in vitro process, whereas the replication is in vivo process, and in PCR it is controlled artificially and is utilized to obtain millions copy of DNA per requirement, as per requirement, whereas replication is the biologically controlled or regulated process in which DNA doubles in S phase before the cell division. So it's not like that it will go for as many copies, but there are checkpoints that at a particular time only it will divide. Now in PCR only target DNA divides, okay, whereas in this whole DNA divides because cell has to replicate. PCR is discontinuous process as compared to replication, which is continuous, and PCR uses DNA primer where it uses the RNA primer which are synthesized by primase. PCR, the thermophilic DNA polymerase such as Taq polymerase is used here. Normal, you can say polymerase is used. And PCR, no replication fork are formed, whereas in replication, leading and lagging strands are synthesized by formation of replication form. So here you gain much better understanding of PCR. Thank you.